From the Donald Danforth Plant Science Center, HEC-TV, Boeing, and the Danforth Center are proud to present Conversations, bringing plant science discoveries to the marketplace.
What a great crowd. What a terrific group we've got here today. Hello, my name is Jay Noose, and I'm the chair of the Friends Committee of the Danforth Plant Science Center. I'd like to extend a warm welcome to each of you to our center, especially our esteemed leaders, Bill Danforth, Jim Carrington, and Jim's lovely wife, Terry. And a very warm welcome to our center friends, and especially their guests who may be visiting the center today for their very, very first time. We welcome you. I'd like to say thanks to our Friends Committee for hosting this Conversations program, and to Molly Klein, chair of our Conversations Committee, and the rest of our committee for picking today's timely topic, bringing plant science discoveries to the marketplace. Now I'm going to pause and go off script here for a second. We got you here with that title. What we're really going to be talking about is RNA interference, or some would call it RNA silencing, or RNAi for short. And they figured a title like RNA interferencing would have gotten a very, very different crowd to show up tonight. So thank you for coming. I'd like to thank Molly's committee for developing this topic, identifying our panelists today. Today's program marks the 10th anniversary of our conversation series, 10 years. That's terrific, a happy anniversary to about 50 conversation series. So we are very grateful to the Boeing Company, the sponsor of our conversation series for the past 7 years. Tonight's conversation program would not be possible without Boeing's generous continuing support. As a part of our partnership with HEC-TV, we are pleased to note that this evening's program is being recorded. It will be aired on HEC-TV multiple times on Sunday evenings at 5 p.m. beginning September 15th. I've got to make sure I look okay. You can also access this and past conversation programs on the Danforth Center website. So without further delay, it is my great pleasure to introduce our moderator, Vijay Chauhan, Senior Entrepreneur in Residence at the Biogenerator, and our distinguished panelist, Dr. Jim Carrington, President of the Danforth Plant Science Center, and Dr. Tom Adams, Vice President of Chemistry Technology at Monsanto. Tom is going to do particularly well tonight because his wife is in the front row. We welcome Erin Bell. Thank you. Panelists.
Thank you, Jay. Good evening, everybody. Thank you so much for coming out to the center on a warm, hot day. We're going to make it worth your while. I am so happy to be here with Dr. Carrington and Dr. Adams. I'm going to call him Jim and Tom. And the first thing that should be very obvious to everybody here is that I'm not Jim Davis. Your moderator for the last 10 years, for the last 49 of these. But what is not probably known to you all is that I am Jim Davis's Irish brother.
These are some big jokes.
Even he doesn't know that. So thank you for giving me a chance to do this. The topic, of course, is a very important one, bringing discoveries in plant sciences to the marketplace. We will talk about that in the context of a very exciting technology, RNAi silencing. But before we start there, I would like to just ask Jim and Tom, you guys have been working across the street for a few years now. How did you get to meet each other? Was that through interaction across the street? Can you just tell us a little bit about how you know each other?
I've never seen him before in my life.
That was my line. We've known each other since before we knew what RNAi was.
Yeah, before it existed.
No, I think it existed before it was discovered.
Yeah, we didn't know about it. Nobody knew about it. 24 years.
24 years, is that what it adds up to?
I think so.
Yeah, we met over pizza and say, well, I think that's how we got to be friends. I was interviewing for a job at Texas A&M where Jim already was, and actually had brought Aaron down for a second visit to decide if we wanted to go, and we weren't too sure. And then Jim invited us over for pizza, and we decided to stay and had a pretty good time there.
Sealed the deal.
Yeah, all it takes is pizza.
Actually, there is a side note, and I'm being serious here. There was an important lesson that maybe wasn't immediately obvious, but I came to appreciate over time. If you want to recruit somebody, be nice to them. And it does make a difference. The little things do make a difference, like taking someone who doesn't have a dinner and giving them a piece of pizza.
It wasn't really good pizza either. It was just
College Station pizza.
So I think we're going to silence this and move to RNAi. Thank you, Jim and Tom. As you can tell, these guys know each other for some time, brilliant in their world, their work and their fields, particularly in this field of RNAi silencing. You're seeing in Jim, one of the leading researchers, and in Tom, a leader of a phenomenal commercialization effort in this area. So I wanted to start off by asking a question. We're talking about bringing plant science discoveries to the marketplace. So Jim, for Danforth Center and its mission, what does marketplace mean? Can you define the marketplace? And then how does that marketplace and the definition of its mission affect the agenda, the research agenda?
Mm-hmm. Well, that's a good question. It's kind of a broad question, but just to remind everybody, the mission of the Danforth Center is to do good things in the world using plant science, and that means improve the availability and quality of food, improve the environment using plants, improve the environment around agriculture, and improve the bioeconomy here in St. Louis. And so when we talk about doing work and a marketplace for that work, it means a couple of different things. First of all, the work that we do around food, the marketplace for us is actually sometimes not a marketplace at all. It's smallholder farmers. It can also be commercial marketplaces if we license a technology. We don't do what Monsanto does in terms of breeding new corn varieties, but we do get involved in improving staple crops. And so the market there is largely smallholder farmers. For the bioenergy research, for example, the marketplace will be companies that can take a discovery we do here and commercialize it. So we'll license a technology. So those companies are our marketplace. And then for the bioeconomy piece, the marketplace is St. Louis. We spin companies out of the Danforth Center, but we also license technology to companies here. And we try to recruit companies to the area, to Bridge Park and elsewhere in St. Louis. So the marketplace for that piece are the companies, the institutions here, and they're all partners with us.
Thank you. Tom, how about that for commercial enterprises like Monsanto and peers of yours in the agribusiness industry?
I think it's an easy question to answer, especially following Jim. I think that it's interesting how aligned what Jim described for an institution like the Danforth is with the commercial interests of a company like Monsanto, because we are a company that's all about agriculture. It's 100% focused on agriculture, and it's a great time to be associated with agriculture because there are so many interesting global trends that are associated with agriculture when you think about food security, the needs of feeding a growing population, energy, all sorts of things that are converging on agriculture and the need to produce a lot more, whether it's from large farmers or smallholder farmers. And I think it comes together in many different ways in Monsanto. And we, a few years ago, laid out a challenge that's very similar to the Danforth Center's mission, but because we're a company, we like to put numbers on it too. And we said, if you look at the growth of the population and what's necessary, that we want to double the yields of all the major crops that Monsanto's a part of, not by ourselves, but with the help of others by 2030. And we're on our way to doing that. I think there's a long way for all of us to get to that point, but it's pretty important that we get there. When there were some numbers that I was looking at earlier today about the amount of growth that's necessary to just keep up. And in the next 10 years, we need to be in a position that we're producing, it's about 7 billion more bushels of corn and soybeans than we do today, which is a couple 100 million acres if you keep yielding the way we do today. So those are really big numbers and a big challenge to do.
Just to keep up, we need to increase on a year-by-year basis of about 1.75% global productivity. And we're right now at about 1.25% increase in productivity. So that gap, if you graph it out, gets quite wide after 30 years or at 2030, unless we figure out ways to close it.
So you can see the missions are pretty aligned, working on the same big issues that affect us. And what's very interesting, and we'll be talking about it today, this technology called RNAi silencing is being used by both organizations, and how they are using that to bring dramatic change to address this problem that we were talking about. So talking about plant science discoveries, Tom, what are the top 3 plant science discoveries in the last 30 years, and where does RNAi silencing fit in that? Top 3, top 5?
That's an interesting question. I think if you 30 years, I'm going to maybe cheat just a little bit on the edge of that, but I think would have to say, and I'm going to limit 30 years. It's things that I've been part of or seen, and the ability to transform plants has clearly been a major undertaking, both for research and for making some crops that have had a really big impact in agriculture. So I'd certainly include that. I think when you think about the science that came together around transformation and some of the work that's gone on since then in genomics, and what that's done to enable breeding and the ability to improve crop productivity faster through breeding using genomics technologies is sort of a big one, but I'd call that a second. And then I think I'd put the third would be RNAi, and it's yet to really be shown how big RNAi will be in terms of how it affects agriculture, but it's a really huge scientific advance in understanding a lot of things that are going on in plants.
Thank you. So I've got a question for the audience. What is common across NutraSweet, the sweetener that you use in Diet Coke, Splenda, which is another sweetener that you use, Viagra, woke you up, didn't I?
Another sweetener.
And RNA, another sweetener, Tom says.
Going blue. We're going blue now?
And RNAi silencing. Anybody venture to guess? What's common across them? Well, what's common across them is that all of them are accidental discoveries. So with Splenda, it was a chlorinated sugar being developed for an industrial application. And the research head said to the junior scientist, I want you to test something. And this guy heard it as, I want you to taste it. So he puts his finger in there and he tastes it. And he says, boss, this is sweet, really sweet. An unexpected development. So we all think about science as a very ordered, disciplined, sequential process. You have an idea, you have an experiment, you're hoping for an outcome. Well, science doesn't quite work like that way. And RNA silencing has a similar journey. So with that, Jim, could you tell us a little bit about RNAi silencing and sort of how that phenomenon was discovered?
Yeah. So I had a sense you were going to ask that question. And generally, we do not have props during conversations. And I made an executive decision to have a prop to answer this question. So if Bill can, it's up there now. There are 3 images up on the screen that will help tell this brief story. In about 1988 or so, there was a biotech company out in the San Francisco Bay Area called Advanced Genetic Sciences. And they wanted to take this, what Tom categorized as one of the top 3 big discoveries over the last 30 years, the ability to make transgenic plants. They wanted to make ornamental plants that had interesting color patterns and interesting different colorations. And so they found a gene that made petunias purple. So you've all seen petunias. There's a little bit of purple pigment in the flowers. They wanted to make super purple petunias. And so they took that gene and they added extra copies to petunia, because if one copy is good, more copies will make more pigment and they'll be more purple. They wanted to get the plants that were on the left up there. But the striking observation that they made, and others followed it up very quickly, was that many of the plants that they got in their experiments did not look like the ones on the left that were super purple. They looked like the ones in the middle and on the right, where parts of the flower made no pigment at all. In some cases, there were these star patterns that were really striking. I think ornamentally, the one in the middle and on the right are fascinating, and I think some of them have even been attempted to be commercialized. But they, after doing this and observing these really strange flower patterns with white sectors, they could not figure out why, if they were adding extra copies of the pigment gene, much of the flower was white. It made no sense, and there was no explanation for it. The literature was searched, colleagues were consulted, there was no good explanation for it. And so scientists took this on as a basic fundamental problem, asking why in these experiments some of the flowers are white. Did it have any practical, was this going to feed anybody? No. Was there any important biological insight to be made? Maybe, but it wasn't obvious. But there was a lot of curiosity about it, and talented scientists took it on as a challenge to figure it out. It took them, gradually over the course of about 8 years to figure out why the plants looked white sometimes. And the reason was because of RNA interference, or what was termed by Craig Mello, who won the Nobel Prize in 2006 for making one of the key discoveries for how this works. What was eventually discovered collectively by the community, we played a little small piece in that, was that plants have a system to turn genes off if there's too many copies. This is what this small biotech company was trying to do, and they, by total accident, stumbled onto this mechanism. Our little contribution, and I'll end with this, was to figure out why plants do this. Why would you want to turn genes off if there's too many copies? You might have some intuition about it, but it turns out, and Hernan and Alberto, who were 2 postdoctorals in the lab, had some displays out here to explain it. It's one of the ways that plants recognize viruses and other invasive entities like viruses, and it turns the viruses off. This was an antiviral immune system that these scientists stumbled across, and they visualized it with their pigment experiment. That was one of the more important things we ever did, to figure out that this is a natural process that has a real function in nature, and it's to protect plants against viruses.
Thank you, Jim. So you can see, as in life, it's also in science. When life gives you lemons, you make lemonade. And here's a great example of what scientists, resourceful scientists do. They take an unexpected result. Instead of hitting a roadblock and saying, I don't know, they start to discover this hidden answer, and up comes an innovation like this. So Tom, can you tell us a little bit about examples of making lemonade out of lemons that you have in this field of RNAi silencing or biotechnology at Monsanto or other industry?
Sure, I can tell you a story, maybe to build off the story that Jim just told, and I think it's actually, we haven't made the lemonade yet, but I think we're actually maybe working on something a little bit more like a fine scotch than lemonade. But
there you go.
A very similar type of phenomenon to what Jim just described occurred in a weed that some people may have read about in the New York Times and things that gets called a superweed. It's a weed called Palmer pigweed that's really been a devastation to cotton growers and now moving into soybean fields in the South, where it's a weed that can grow a stem about this big if you let it grow all the way through the season, and basically you can't drive a harvester through. People go in with machetes to cut them down. So you really want a herbicide to take care of that. And we have a very good herbicide called Roundup, and we have a product we sell called Roundup Ready Soybean, Roundup Ready Cotton, and been a very, very phenomenal product in terms of how it's penetrated a lot of area. But nature is pretty powerful, and when you use any kind of thing like a herbicide like that, you'll find that there are examples of things in nature that have found a way to be resistant to it. And this Palmer pigweed did exactly what the scientists did with the petunia. It actually took the gene that encodes the protein that glyphosate, the Roundup, causes to be inhibited when it kills a weed, and it made a whole bunch of copies of it. So it made like 100 copies, but rather than turning the Palmer pigweed purple, it made Palmer pigweed so it was resistant to Roundup, so it was Roundup Ready tolerant. So a scientist in my group was studying this phenomenon and trying to understand it, and he had kind of a wild idea that maybe I could spray RNA on to the plant, and I could do that in conjunction with Roundup, and I'd cause the transcription of that gene to go down just as though it was a virus, and I'd make the plant sensitive to Roundup again. And if he'd asked me if he should do the experiment, I of course would have told him no, that's crazy. But fortunately, usually scientists are smart enough not to ask us for advice about that. And he did the experiment, and I was walking down the hall, and he said, it worked. And didn't know what he was talking about, so he took me out to the greenhouse and showed me these dead plants. And from that, I think some of you might have seen the displays where we've been able to look at other ways in which spraying RNA on a plant can have profound effects on the phenotype. And it gives you a whole different way to think about how some of the new technologies can be used in very positive ways, and potentially reducing the amount of herbicide you need to use, insecticides, and other types of treatments. So really exciting new kind of technology coming out of a resistant weed.
So what I learned from that is when life gives you lemon, make fine scotch. Don't just stop at lemonade.
Just as a side note on this, there were actually a lot of crazy scientists who tried to do this experiment, and it didn't work because they didn't do the experiment properly. Just kind of confirmed they were crazy. But
and I'd heard about them, which is why I would have told them not to bother.
Yeah, sure.
So you hear again and again these stories of great scientists who hit a brick wall, come up with a dead result, and some take it to the next level, some of them don't. So Jim, can you share with us what the real world of science looks like when you hit this brick wall? Any personal stories, any examples of an unexpected result, not what you're looking for, you had a great grant, they were looking for some wonderful results, but you're not getting anything like that. Now what do you do?
Yeah, well, that's another good question. Maybe I should explain what RNA is.
Yep.
If you know what RNA is, raise your hand very high. Terry, you better raise your hand. Okay, so about a quarter to a third knows what RNA is. RNA is genetic information in all types of living cells. And it's the information in the cell that is relayed from the DNA. So you all know what DNA is. It comprises your genome. You're all a bunch of genes in unique combinations and unique flavors of genes. That's why we're all different. But your genes make RNA, a chemical, that instructs the cell what to do based on your gene. So you don't need to know any more than its genetic information molecule in the cell. And so we've studied RNA for, I've studied RNA since 1982. And what was the question? The question was...
Have you ever had any problems?
No.
So
the question was, what happens if you hit a brick wall or make a discovery that you can't explain? Well, in this case with RNA interference, a lot of scientists went down a lot of rabbit holes trying to figure it out, and a lot of them never came back. All right? The ones who went down the rabbit hole and found something really insightful and came back out with the rabbit. Is that the right analogy? Am I
That's
on
a
the right
good one.
metaphor here?
Just keep going.
Yeah. The ones who came down the holes and up with the rabbits were the ones who had a couple of key insights, and they were scientists who were smart enough and broad enough to pay attention to what was happening in other fields of research. So the plant biologists who made the key insightful discoveries actually had their eye on some experiments and some science that people working in worms like the crawling worms, were doing. Now, why would plant biologists be paying attention to what scientists working on worms were doing? Well, because there's a lot of interesting science being done in worms, and there's a lot of commonalities in the way RNA works in worms and plants. And it turns out that the scientists working in worms found the same phenomenon, that when you added extra genes, they would turn off. And the plant people and the worm people, who were smart enough to be looking at each other's work, started talking to one another and started going to each other's meetings and started collaborating in some cases and asking the right questions and sharing the right information. But they also were the best guessers on how the system worked. And science is just a matter of making guesses, predicting. Sandy and I had a good discussion about this one day, guessing how a biological system works, and then designing an experiment that tests whether your guess is good or not. And if the experiment is predicted by your guess, if the outcome is predicted by the guess you made, you're on the right track. So the really good scientists like Craig Mello and David Balcom made some really good guesses. They were paying attention to each other's work. They were bold enough to quit doing what they were doing in the laboratory that might have been productive, and they changed course because they sniffed something that was really interesting and important and potentially broadly applicable.
So it's a really fascinating story, isn't it? Two camps of people not talking to each other, discovering the same thing, starting talking to each other, and there comes the breakthrough. So given that wonderful background, Jim, can you tell us a little bit about projects going on at the center that are using RNAi technology?
Mm-hmm. Well, so it's kind of a little microcosm of what the Danforth Center is all about. We have work at the center still focused on understanding the basic mechanism of how RNA silencing works. And Alberto, who was at one of the displays, and Hernan, and Sonia, and Agata, and Marian, a visitor in the lab, were out guiding some of you through that. Studying the basic mechanism is still really interesting and very important because what we learn today at the basic level is going to be applied at some point in the future. But we also have work that applies RNA silencing now with current technology to do useful things. Nigel Taylor had another display around cassava. We're using RNA silencing to shut down viruses in anticipation that those plants will become infected in the field. In other words, we're pre-immunizing the plants against devastating cassava viruses like cassava brown streak virus here in the laboratory and out in the field. And we know how to do that now because the basic science was done. And now we're at a point where we can translate it and apply it out in the field. So that's an example of a project where there's not a large commercial market. It's a smallholder farmer market. The market is in sub-Saharan Africa. It's in Asia. It's not in the US. It's not in Europe. But this is an example of a project that will benefit people because crops will be improved through their immunity to devastating viruses.
Well, thank you for that following up on that, there's a really great question here. Since RNA is in all living things, will silencing them be considered GMO?
Since RNA, can you read that again?
Yeah, so since RNA is in all living organisms,
Yeah.
would silencing them be considered genetically modifying?
Yeah, it depends how you do it. So Tom can explain to you how you can shut genes down without being GMO. Now, many of you already know this. A GMO, or genetically modified organism, is a definition if you have a foreign gene that you've added intentionally in the laboratory. So if you add one extra gene, like a gene that triggers immunity through RNA interference, that's a GMO. It's the simple criteria that defines GMO. But Tom can get similar effects without being a GMO. So the answer is it depends how you do it.
So Tom,
Maybe you can explain more on how it works with
tell us about that
BioDirect.
approach.
Well, I think what Jim's referring to is rather than modifying the genome so that the genome, when it replicates, will carry it on to the next generation, we've developed some technology where one can apply RNA to a leaf of a plant, and it will get into the cell, have its effect, but never integrate into the genome. So it's acting much more similar to how a chemical might act, how you're used to a chemical acting.
But it's basically the same type of molecule that the cell makes. You're adding it with an aerial spray, for example. The plants are taking it up, and the plants are not distinguishing those molecules that get taken up versus the ones that are made in the cells, and therefore you can get the effect.
And because the genome of every plant or every organism that we're trying to affect is actually somewhat different in the basic DNA, and this is actually a mechanism, a natural mechanism that uses the exact sequence or order of things in the DNA chain or RNA chain. You can design it so it'll only affect one plant and not another in a very simple way.
And you might be able to do this to inhibit a virus, and in the next year, if there's a new virus, you could conceivably respond very quickly.
Much like a vaccine.
That's right, exactly.
So that's a great example of how this new technology addresses some of the concerns that people had with genetic technology. This will help achieve similar outcomes with a different approach. I concluded from that it's like applying a topical lotion to become Superman versus taking spinach every day. So rather than consuming the spinach, just apply it, and you'll become a Superman. So talking about
Isn't Popeye? Isn't Popeye the spinach guy?
Popeye the spinach man.
It's a mixed metaphor, it's okay. Don't correct the host.
So in bringing plant science discoveries to the marketplace, it's a very big challenge because, as we all know, it takes a long time for the science to develop, convert that into a product, get the regulatory clearance, and then finally gets to the marketplace. So it becomes a very important decision for companies and institutions to make a decision on, do we want to lead in this field, or do we want to follow in this field, let somebody else lead, learn from their mistakes, and then come after that. So give us an example, Tom, of where the company or your projects decided to take a leadership role, why it decided to take that, and what were the implications of doing something like that?
I think maybe to answer the question slightly differently about whether leading or following is the approach, I think the way that we would tend to approach things is if something is worth doing, it's worth leading. So I think when we look at an opportunity and something that could really make a difference, we're not going to be shy about trying to make an effort to make that happen. And certainly I don't like to approach things that way. What we want to do, though, is not, while jumping in with both feet to succeed, also identifying very clearly these brick walls that Jim was referring to and understanding, is this a brick wall that I can get through or not? So it's not so much about whether I want to wait for someone else to figure out whether they can do it, because then it's too late and it's not really going to make as big a difference. It's jumping in and finding out whether I'm going to be able to lead, and if I can't, then stop and move to another opportunity.
And Jim, how about at the center?
Well, you know, it's like those commercials that you see with the guy sitting with the 4 kids. Is it better to lead or to follow? You know, the obvious answer is it's better to lead because it's much more exciting at the front. It's not so difficult to figure out what to do if you're following. You just do what the guy in front of you does. But the impact of your work and you know, the relevance of what you do will be much less because you'll basically already know the answer. So I'm talking about in the basic discovery realm. So in basic discovery, you want to be the person. You want to be the man or the woman or the group that makes something no one else, makes a discovery nobody else knows. Now there's a lot of reasons because that's progress, right? And that's why scientists are in the business. There's also I'll give you a little insight about why making a discovery is so much fun. And the guy who explained this to me was a guy named Victor Ambrose, who is another one of these worm researchers I've known for a long time. And he was one of the key guys that was in on the early days of RNA interference. And we were talking one day about why it's so exciting to be a scientist. And he said, it's because if you find out something that's new, for some window of time, it might be an hour, it might be a day, it might be a week, you know something that nobody else in the universe knows. And that's a lot of fun. It's like you've got a secret, the world doesn't know, and they're going to know whenever it's up to you to tell them.
So you know, we can all...
And then, of course, on the applied side, you want to lead because there's a lot of reasons why people aren't out serving smallholder farmers. There's a lot of reasons why big, tough problems aren't being addressed. It's because they're tough. So you want to be the leader in addressing the tough problems because most people won't tackle them. That's part of the mission here.
And for all of you who've been associated with the center, you know that the Danforth Plant Sciences Center is an absolute leader in all the disciplines that they are engaged in. And now you heard today about their leadership in RNAi silencing. To me, a very dramatic example of whether you lead or follow is realized when you talk to the employees of BlackBerry. BlackBerry was the dominant email device, the smartphone device, and they thought they knew everything about what people wanted with the smartphone until Steve Jobs comes along with the iPhone. And for the longest time, the people at BlackBerry said, nobody's ever going to use that in a corporate environment because people love their email the way we do it. And here we have an example of a company after 4 or 5 attempts, because they followed, they could never catch up. And so you see here 2 leaders in their field exerting great leadership in areas that are pretty untested. And I think this is what I find most exciting about commercializing discoveries. You are in a landscape where nothing is known, a lot of risk. You have to continuously convince your bosses that this money is well spent. So Tom, give us an example of a really challenging project that you were faced with to sell to your leaders at Monsanto that you knew on the face of it there just was going to be a lot of uncertainty and risk. How do you go about doing that?
I think, and I've been talking about what we call BioDirect, which is what the name that we've used to refer to the technology, since it's a lot easier than trying to explain RNA interference all the time, to what we're doing with spray on RNA. And it's one of the great things that I've found working in Monsanto, and I can't say this is true with every company, but if you can have a vision of what could be if we can make all of the pieces work, it's actually not that hard to sell to the leadership. It's and if you have a plan to work your way through whether it's going to make it to that vision, I don't know that I've ever had a terrible challenge selling it. It's hard work every day trying to make it through that plan, but the excitement and the opportunity outweighs that.
So Tom's a great salesman, which means he knows how to establish the value of the investment, what will investors get for it, what your shareholders get for it.
I have a question. In the case of BioDirect, or the spray on RNAi, a lot of the traits that you're producing, whether it's insect resistance or herbicide resistance, are currently being delivered through your GMO crops. And so how do you deal with the potential that one side of the company can obsolete some other products in the same company?
So, first I think that it's very unlikely that one will obsolete the other. I think they each have their place and they complement each other quite well. And I think one of the things you're going to see happening in agriculture more and more over the next few years, and the types of problems that are in more modern agriculture, the types of problems that are being faced are not things that are single solution problems. And each piece of this will bring something that brings together the whole solution. So I think one of the things that we're talking about now is what we call integrated farming systems, which has a lot to do with how you plant, how you decide which hybrid to plant in which part of the field, but it also has to do with which chemistry you spray, which trait you have, which biodirect molecule you might include, or which microbial you might include on your seed when you plant it. So I don't think, I would say, given the uncertainty of which things are going to be the most successful, I don't feel a lot of nervousness that one part is going to obsolete the other. The problem that we're trying to solve, and bringing it back to 7 billion bushels of corn and soybean that aren't produced today that need to be produced 10 years from now, it's a big enough problem that you don't really worry about who wins. We've got to win.
So this is the last question. It's for you, Jim. What is your greatest hope and expectation for RNAi silencing's impact at the Danforth Center? What are you thinking? If it can do X, Y, Z, what's your hope on that?
Yeah, well, the Danforth Center is a very simple organization. We have a relatively straightforward mission. So let's just revert back to the mission and ask, what are we here for? So if we can use RNAi to make crops more productive, whether they be commercialized industrial scale crops like corn and soybean, or whether we can make banana, sweet potato, cowpea, and cassava more productive using RNAi, either because we're protecting them from viruses or pests or something else, changing the oil composition, that's what using the science at the Danforth Center to increase productivity is all about. If we can deliver technology that gets taken up by industry, whether, again, it's a startup or whether it's a mid-size or a big company like the one across the street, and they see value in something that started off as a small discovery here, but through their commercialization efforts and their scale-up and their application, if we see that out in the field in Missouri, that's big-time success. If we spin out a company that's here in 10 years and there's 500 employees, that's success not for us, but for the region. So those are just some of the things that if RNAi is a foundational technology or a piece of the science, that's big-time success.
Thank you, Jim. I don't know about you guys, but boy, he's the right guy for this job, isn't it? Focused on the mission, always on trying, knows what he needs to do. So I wanted to thank you all for coming on this hot evening. Hopefully it was worth your while. Jim and Tom made my job really easy. And just to close out the evening, I'd love to have Dr. Danforth come and say a few things.
Well, let's say thank you very much for our moderator and our presenters. I think it's a great session. And I just want to say I've been to these sessions for 10 years, and I've never heard a more exciting session than this one. People may say, well, you say that all the time. And I would say, I hope I could say it next time, too. But, this has been very special because it's shown more insight into science and how science worked than I think we've ever had before. We've had a lot of discussions about major problems and what people are doing to solve them and so on. But this really got into the basics of science, and we are a science institution. And as I want to think, we live or die on the quality of the science. It's very important. And I was thinking about being first or not. You know, there are natural human responses. Depends on the era in which you're working. But some of them have been quite dramatic. Galileo was penalized, had a terrible life after he made some discoveries that weren't accepted in his day. When the Johannes Kepler figured out the motion of planets, he was so excited with what he'd figured out. He said, he was a religious man, he said, "Oh God, I think thy thoughts after thee." So that is one response. And for someone looking at it, one of the greatest scientists in history was Isaac Newton. And he was so impressive to the poet Alexander Pope that Alexander Pope said or wrote, "Nature and nature's laws were hid in night. God said, let Newton be, and all was light." So thank you for making it all light.